US.
Right.
So then some of, even my father was a little bit hesitant. He was government job. Government job means kind of secure. And that was a class 1 officer post. But then again I took risk. I said no, I haven't done biochemistry to do this kind of job.
Yeah.
So I want to, when I come back that is a different story, but at least I want to go and learn the advanced research in biochemistry. See, those days I worked one of the best lab in India those days, but still in biochemistry, especially molecular biology, cell biology, we were very naive in the whole infrastructure.
Right. It was still growing.
Yeah, it was still growing. So when I went there, I realized how much advance has happened. And that's where I learned molecular biology, cell biology, which is essential for today's biopharmacy.
Yeah.
So then I was doing work on the cancer research, metabolic disorder and all this. And then I came back because that's for my family reason. I came back here and started, most of the people in India come back as a pool officer in CSI.
Right.
And then they slowly get department. I came straight to an industry, which again something new those days.
Yeah.
Very few people. Nowadays a lot of people come.
Yeah.
Those days are very few people because there was not much industry job of that kind of.
Correct.
So I came and I started drug discovery. That is again, I will say the great person, Dr. Ranjan, who took that.
Reddy's founder.
Founder of Dr. Ranjan, that he will do drug discovery.
Wow.
Most of the people used to say, they used to actually joke with us.
Yeah.
You know, Dr. Reddy made a lot of money with generics and all that. So now you want to waste some money. Actually, that was the thought those days.
Those days, yeah.
But we proved ourselves, and that is very great for Dr. Reddy's. I mean, I used to work, I have my direct bosses there, but he used to call me every alternate day. And call me at late evening or early morning. He might be then. Sometimes I used to hear he is painting when he is talking to me. I said, what happening? No, I am doing my morning walk. I am in Hyde Park or I am in Central Park in New York. So he might have met with somebody, some head of the organization. And he got some ideas.
Yeah.
So immediately next day morning or evening he will call you. And then we will discuss.
Yeah.
And that even when I left Dr. Reddy's, I used to meet with you every month for a lunch.
Yeah.
And we used to discuss whatever new things to research and whatever he is hearing from people. So that way, that's even if I didn't work at Dr. Reddy's, I have a lot of. I brought him in after even at USP. I took my CEO, global CEO, to him, and he met him and all that. So that way, that's a great interaction, and that's a bit, as I learned at US about the science, and also because that's another guy I have a lot of respect. My boss in US, he was a big guy in metabolic disorders, cancer research. So I learned a lot of things about those areas from him. When I came to India, I learned this industry and industrial research from Dr. Lee. These two people are, I have a lot of respect to me, who have to some extent guided me in my future. Then of course, I worked at Dr. Reddy's for discovery research. We were lucky that we were the first one to out-license molecules to Novo, Nordisk and Novartis, and also put it to the phase 3. But then again, that is a big disappointment that both the molecules failed, not for clinical. They were excellent in efficacy in clinical, but in a long term, in clinical trial, when you do for an NCE, new chemical entity, you have to do parallel long-term toxicity. In about 6 months toxicity study, there was some safety issues were raised, and metabolic disorder is something where you have to take the medicine for life. Yeah. So it is very strict as far as the safety issues are concerned. In cancer, because that's a kind of death or living status. So, lot of toxicity, because the drugs itself, cytotoxic drugs have a lot of toxicity. So, little bit of toxicity is allowed. Even in antibiotic, because that's a short term. Even if you have a little bit toxicity, when you stop it, it will come back.
Yeah.
but not in metabolic disorder, cardiovascular and all these.
Right, right,
So it gone there.
right.
Then from there I moved to GDK Bio, and then it was another interesting experience in a CRO.
Yeah.
Because those are the early days
Of the
when the CRO business started in India, especially in biology.
Right, right.
So it was great of Sanjay Reddy
Oh, okay.
and Brar,
Mhm.
Mr. Brar, who was under Maxi.
Mhm.
They were the one who started this.
GDK.
So yeah, and so that was again another learning from Mr. Brar.
Mhm.
He was another pioneer guy in India in this pharma research and all that.
Okay.
Because he was the main pillar of vaccine.
Okay, okay.
So that's again another thing I have learned from there, and we did pretty good as there. And there also I continue to work with the MNCs. That is a big advantage from Reddy's to here. I mean, I left US, but I both the places I work with all MNCs.
Yeah.
And they are based in US or Europe.
Yeah.
So that contact was there.
Yeah.
And so I never felt that I am not able to see what is happening in those countries.
Yeah.
Because I am meeting them, I am getting updated,
Right.
and they are also telling, okay, you do this way, you do that way, like that. Yeah.
Right.
Then I moved to US Pharma.
US.
That time actually US Pharmacopoeia, that is a cult kind of.
Yeah, USP.
That is a 200 years old organization.
Yeah.
I mean, you can't imagine in
Yeah.
US. Right. Yeah. And they are the kind of, many people told me, USP, that is the Bible.
Yeah. Everyone study that, yeah.
Yeah. All the guidelines are put
Yeah.
in. So when I got that opportunity, then immediately I jumped. That's such a renowned, reputed organization across the globe.
Right.
So and they were setting up their lab.
Huh.
So in Reddy's also, I set up the biology.
Yeah,
and
you are the first person to do it.
So everywhere I have done it. In GDK also, I didn't set it up, but I expanded this thing.
Yeah.
So here from the beginning we worked
Yeah.
together, and then we set up the whole lab and all that. And then not only here, in USP I actually set up lab in China and Ghana also.
Oh, okay.
And my early part of USP, I was actually overseeing our US lab, India lab, China lab and Ghana lab.
Okay.
So there was a lot of, you say, global interaction.
Global
This
interaction.
part, and I used to go there.
Yeah,
meet
traveling was so much,
stakeholders. But my travel was more on my next part. In the second part of USP, I worked Dr. Reddy's for 12 years.
Right.
I worked at USP for 11 years.
Oh, okay.
And then I retired from USP.
Yeah, yeah.
So in the second part actually, I came out from the lab.
Right.
And I focuses on that because that time USP also expanding their activities in different part of the world. So I was they call it global vice president of the scientific reach out. So there, what I had to do is, we have our new team coming up for Southeast Asia, stationed at Singapore, who are looking at Korea, Japan, Singapore, Indonesia, Thailand, all these areas. And Korea, Japan, and for your information that my research in Dr. Reddy's was on NCs, new chemical
Entities, yeah.
whereas in USP is totally on the biopharma.
Oh, biopharma.
The whole thing is on biopharma. There was a different group working on chemical medicine was the traditional USP, 200 years.
Yeah.
But biopharma is the new what USP was doing.
Oh.
And I was involved actually building strategy and all that with my counterpart at US for the USP.
Right.
Yeah. So these are the things which I am very satisfied everywhere I have started and make some impact also.
Impact and difference.
So from USP when I did, I got that huge opportunity to actually I have crisscrossed the whole China.
Yeah.
And met with the regulators, with the manufacturers, went to Korea, went to Japan, went to Singapore, and these are the places where all the innovations and new thing happening in Asia.
Yeah.
Of course, it's in US and Europe. So that's an excellent opportunity.
Yeah.
And that gave me a lot of things. Then I realized where we are lacking in India and where we should have to think, and that I used to mention in all of the government meetings and even now also in different committees where I am there. I mentioned them. So these I have seen. So we have to do it here, really we have to reach there.
Yeah.
Because they have gone way ahead of us.
Yeah, right. So I have one more question, sir, because you started working with NCE. After that, you started working in USB and biology. So now everyone is talking about one most important thing, that biologics is something which is the future of the medicines. And everyone is speaking about discovery of biologics, how the discovery and how trials and all. Can you put some light that what exactly is biologics and what makes it different from NCE and what is the future of this biologics?
Yeah, let me go step by step.
Yeah.
So, a biologics in very simple term is a drug which is produced in a living organism.
Oh.
Whereas a chemical entity is a drug which is synthesized, drug in large scale, of course not synthesized in labs, in reactors, but that is a synthetic drug.
Synthetic drug.
Whereas this is a in a living system.
Living
And that's where the major difference and the challenges come.
Okay.
The chemical medicines are actually comparatively small molecules. Even if you look at the most complex or the large molecule in a chemical medicine and compare it with the simplest biologics, there's a huge difference.
Oh,
Yeah.
okay.
So that is the one part. Second part is, in a chemical synthesis, it is totally under your control. You synthesize as you want and all.
Right.
But in a living system, that system has its own mind. Whatever you do,
Yeah.
you do manipulation with
Right.
molecular biology
Right.
and all that,
Right.
put that thing there. But when it makes actually, it makes on its own.
Yeah.
So always there will be some difference.
Mm.
And then the issue is that, I will come next stage.
Hmm.
When it comes there, it became a very complex one.
Yeah.
That is the major challenge there.
Yeah.
That in chemical, that one is synthesis and living. Second is the structure ones. Small molecules are much simpler.
Simpler structure.
These are very large and more complex.
Complex.
Here from insulin is the simplest,
Hmm.
and monoclonal antibody and these are the complex.
Okay.
More complex.
Okay.
And the third one is, of course, is the cost. See, as you understand,
Right.
as these molecules are synthetic and simple chemical molecules, they are comparatively cheap and much cheaper from the biologics.
Right.
Biologics, you have to go to a living system and they are complex molecules. So your production, your analysis, everything is much more complex.
Right.
So these become very costly, and that's why I will come to the next stage, how you can make this affordable.
Yeah.
And the third is, of course, in small molecules, generally they are not immunogenic. But whereas in large molecules, these are always immunogenic. Now the question is it a safety issue or it is not? That you must check for all biologics. That's an additional burden when you develop a biology molecule. But that's how it is. Now the question is, as I said, that these are costly. So, see, in India, in generics, we are one of the leaders in the world.
Yes. In
Because economically, this is the way that you make cheaper drugs.
Yeah.
So in biologics, that is called biosimilar. Now you look at the name, biosimilar. I'm not talking bio-identical.
Yeah.
That's the challenge of the biologics. Because I say it is a living system. So it is not under totally your control. So you are making something which is like generic, which is similar to the reference product.
Right.
That means the originator molecule.
Right.
Who discovered the originator
Right.
molecule. But you can never make an identical thing because this is a living, it is made in living system.
Right.
That's why it is called biosimilar.
Biosimilar.
So, when you talk about that, economically it is same as generic. And the purpose is also same, to make affordable drug.
Right.
But the whole development of biosimilar and proving of biosimilar is very difficult.
Very different.
Because similarity is, you cannot avoid little bit dissimilarity in the whole molecule.
Right.
So that you have to accept. That's why you said similar. And the terminology a biosimilar is which is largely similar
Yeah.
to the reference product.
Reference.
The areas of the molecule which are not directly involved in the safety and efficacy and quality of the molecule, there might be a little bit different.
Variation will be there.
That is acceptable. But the areas where you have, which are involved directly with the efficacy, safety and quality of the product, no difference is acceptable. So a part of the molecule has to be identical, but a part is acceptable that it can be different. That's why total you said these are similar.
Very similar.
And that's why when you develop and also in biologics you have different product class.
Right.
And the first is I will say vaccine. Everybody, all of us know the Indian vaccine industry, what they have done in the pandemic.
Yeah.
And then you have recombinant proteins, which actually little bit simpler, like insulin, like filgrastim.
Right.
Then comes the complex monoclonal antibodies.
Yeah.
Okay, like rituximab, bevacizumab.
Yeah.
And then the new ones, which are the cell and gene therapy.
Right.
So maybe today or afterwards I can
We can talk more about it.
cell and because this is the advanced one.
Advanced.
And that's why biologics are so important. Biologics are much less toxic than the chemical molecules. Biologics research are giving this kind of new therapies, such cell and gene therapy, which is treating the most I will say dangerous diseases where we have no answer with the chemical material. So this is the one why biology is so important because it is giving us new kind of medicine which help us to treat the untreatable disease.
Right.
But the challenge is in the development.
Right.
Yeah.
Right. So now I think most importantly that the drugs which were earlier was not being given treatment for most dangerous diseases and biologics has got that answer. But to make a copy of a biologic to a biosimilar, it's challenging. One more.
It's a challenging. Maybe let me explain a little bit of that, how exactly we will do it. Because see, again I'm talking, these are biosimilars.
Biosimilars.
So, and one good thing is, when biosimilar started, it was very difficult. Because see, you need a very sensitive analytical tools to characterize this. These are not small molecules. These are large molecules, and it has that complexity of primary structure, secondary structure, tertiary structure, because it folds and makes the final tertiary structure. So, but one good thing which has happened in last 10 or so years is that the vendors who make the instruments, they have also developed a lot. And so we have really now tools, analytical tools, to study in details or in depth this biological model. So that gives us the tools to really analyze and characterize these biological molecules and see how it is with the reference product.
Right.
Now the base of this is a term called critical quality attributes. Now quality attributes is any specification of a biological molecule which if it changes, it can change the property of the molecule.
Right.
Now in critical quality attributes means those quality attributes which are essential for safety, efficacy and quality of the molecule.
Right.
So when you develop for any product, first you decide which are the critical quality attributes.
Yes.
And how will you measure? Now you know all of this. That how will you measure this? What kind of analytical tools you require, what kind of test methods you require. USP has a lot of guidelines on that aspect. And FDA also has that, WHO has that, EP has that. So then first you go for the analytical detailed characterization to show these molecules are similar. The reason I am spending so much time on analytical characterization is that actually tells you or the regulator that how we will proceed for the clinical development.
Right. Clinical development.
If the analytical characterization is very clear and regulator is happy that yes, you have shown similarity without any question, then your clinical trial design is little bit shorter.
Right.
If it is not, then you might need an extensive clinical trial.
Oh.
Yeah. And you know very well that a clinical trial is the costly part for
Yeah.
and and the biosimilar you are making to make it cheap. So if you have a big clinical trial, you spend a lot of money,
Yeah.
that might affect you to make it cheap.
Yeah.
So in that aspect, if that is very clear, analytical one, then you go, then they say, okay, now you develop the clinical trial,
Design.
then there you do the comparison.
Right.
Yeah, first is the safety and efficacy comparison.
Right.
And that generally you have to just remember, more or less similar population you have to take which has been
Used for biologics.
No, reference product.
Reference product.
Reference product means the originator.
Original product.
Whose biosimilar you are making.
Yes.
Yeah. So that one you have to take both of them and accordingly select which population you will take. You have to look at the gender distribution, you have to look at the comorbidities of the population and accordingly you do. And you check the similarity, do the efficacy and safety study and then produce it there. But there is one unique in biologics in clinical trial, that immunogenicity.
Okay.
Now it is a little bit preclinical side, you can get some idea, there are some in vitro tools, you can check that immunogenicity. But the major immunogenicity is still in the
Clinical trials.
Now there again, if your reference product, biosimilarity is of course,
Yeah.
if it is very good, then people say, okay, it must be same immunogenic as the reference product.
Okay.
Then your design will be very
Yeah.
shorter. But it also depends on how severe was the immunogenic reaction of the reference product. Again,
Oh, okay.
for everybody's understanding, reference product means the originated product.
Yes,
Okay?
yes.
Which is there in small and
Right. The patented molecule.
Right.
Yeah.
If the reference product was showed severe immunogenicity, then you need a extended immunogenicity study.
For biosimilars.
Biosimilars also.
Yeah.
If it is not, generally you can do a short immunogenicity
Short
study
design.
and then continue with post-mortem.
Yeah.
Remember that you have to do.
Do it.
Yeah, because you don't know long term your biosimilar,
Correct.
how it will be. So that is there. But by that time you have marketed the product, you are getting money. By selling
Yeah.
product.
So on a nutshell, it is like when you have said NC, it was a BAB studies or bioequivalence
Right.
studies, it
Very
was
easy.
being done. Very easy. Pharmacokinetic parameters and no disease population, nothing, just copy paste or patent generic. But now biosimilars, when you are saying, it's a very complex to do it.
Because
And
you have to go to the efficacy.
efficacy, yeah. So, but in BAB, it was no efficacy was there. Now it is, you have to test for efficacy, safety, immunogenicity, if it is more dangerous in patented product.
There is another issue.
Yeah.
That is called interchangeable biosimilar.
Oh, wow.
Yeah, because see, we all aspire at the end of the day to go to US.
Right.
Because that's the largest
That's the
market.
market.
Whether small molecule or large
Market, we have to
Yeah.
go to US.
So this is very unique for US. Nowhere else in the world, this interchangeability or interchangeable biosimilar word is there. Anywhere else, a biosimilar means interchangeable.
Okay.
But in US, as in one unique thing is there, the pharmacists who are in like CVS or Walgreens, they can switch. The doctor prescribes maybe the original name
Okay.
of the product, like
Yeah.
rituximab.
Ah,
Okay,
ah.
but the rituximab, the generic name of the Ritux originator and the biosimilars are different. Okay, so a pharmacist, they give that like generic. Generic also the pharmacists they choose which one to give. Now biologics are complex. So FDA doesn't want to take any risk. They say is that you have to do a clinical study to prove that your biosimilar is intact. Scientifically it is the same biosimilar.
Correct.
It is the clinical trial
Right.
where you need to prove. They
Yeah.
said you need to prove in clinical trial that it is interchangeable. So what there are two trial designs. One is called directed direct and another is the integrated.
Okay.
Direct means there is a initial period where you use the reference product and then you have two arms. One arm reference product continue
Yeah.
another round, reference product and the interchangeable biosimilar, you
Okay.
switch.
Yeah.
During the trial, you switch these 2-3 times. And end of the trial, you see how is the safety and
Yeah,
efficacy
efficacy.
and other parameters,
Yeah.
immunogenicity and everything. If there is no difference, then you can claim it or FDA give you interchangeable biosimilar. There is another is integrated where at the first stage itself, there is a reference product, there is an interchangeable biosimilar product. So you check the biosimilarity there itself, these two. Then one stays as reference product and the other one begin the where you switch the interchangeable and the reference product like that. So after that, so you see this is a totally different clinical trial.
Yeah.
So it adds you cost if you go to US for that one. If it is not interchangeable, if you get only biosimilar, then pharmacist can give you there.
The
Then
other molecule,
your
yeah.
market
Will decrease.
penetration will decrease.
Yeah, right.
So that's another challenge,
Yeah.
which is only for biology.
So now there is a word also that interchangeable biosimilars where pharmacists can interchange if you submit this data. If you conduct this kind of clinical trial, then only US FDA will approve it.
And in US, as most of the things are given by, see, doctor writes a prescription.
Correct.
Then it goes to those
Pharmacist,
pharmacists
yeah.
in CVS
Right,
or
right,
in all
right,
those places,
right.
and they decide which one to give.
Which one to give?
So that is very important for your market penetration.
Yeah. So, but the very interesting story is that we MCEs were just a normal BAB study, but now it is clinical trials on complex terms of efficacy and safety and also interchangeability. So now one more interesting question I have, sir. Do you think that India is doing some research on biosimilars now, or in future it can grow? So what do you think?
Yeah, biosimilar, see, I mean, what happens is during COVID period, we have done so much excellent work in vaccines. That the other areas can develop. But biosimilars, India is doing a lot of good work.
Okay.
But still, there is an issue there. I'll tell you that.
Yeah.
See, if you look at the money-wise, financial, if you look DBT's the bioeconomy report of India in 2023, bioeconomy overall, right, which includes biopharma, vaccine, cell gene therapy, agri, diagnostics, all these together. Actually, when in 2014, it was I think around $10 billion or something. Last year, 2023, it was $137 billion.
Oh.
You can understand how much growth has happened.
Right,
Okay?
right.
And out of that, about 49 billion is actually this vaccine, the biopharma. That means mostly vaccine and biosimilar.
Yeah.
Biosimilar, I think about 16, vaccine is 14, and the rest is the CDMO and all those things who are working for others in the biopharma area. And so you can understand there's a big amount of biosimilars which India is earning.
Yeah.
But if you look at that, there are some issues on the quality part.
Quality part.
Because India has about 100 biosimilars in Indian market.
Okay.
But if you look at outside, there are very few of them in the developed markets. Many of these are gone to the what we call global south, those areas. But the developed market, that is Europe and US, Europe still few has gone. US only by is the one who could penetrate US so far, but that is also in collaboration with Maya.
Okay.
The reason is that the 100 biosimilars which are there in India, there is a perception that their quality are not of global standard.
Oh.
Yeah. So
There's a perception.
There is a perception, and at the early days of biosimilar, there were some issues on that, and that's not in India, even in China. I went to China, you know, one of the biosimilars which they approved. Afterward, it has been found out that it was not proper. It was not good.
Yeah.
So then we draw that only. In India also it happened early days, but now regulators. That's why regulators' role is very, very important in this new area. because these are complex.
Right.
So regulator need to understand the complexity of these molecules.
Right.
Thereby, when they approve this,
Right.
they have to ensure that the quality of the products and the similarity they have reviewed properly
With reference.
with reference standards.
Right.
But I am hopeful that in India it is very good, and we are earning about $16 billion with biosimilars.
Oh.
And the opportunity there is two. One is globally, biosimilar market is actually going at a 24% per year. And then there are 10 blockbuster biologics medicine which are coming out of patent in maybe next few years.
Oh.
So this gives a big opportunity
Right.
for the Indian biopharma companies,
Right.
because now there are a couple of companies who have built that capability and the capacity to really go there. But they have to ensure that they are making the products which are of proper quality, working closely with the European authority and the US authority,
US authority.
and see to it that they develop their products as per the regulatory requirements of those countries.
Yeah.
Because if you want to make money, and in that instance I will say our government's vision of bioeconomy in 2030 is 300 billion US dollars.
Oh.
From 137 to 300 billion.
Yeah, double.
Next 8 years.
8 years.
So for that reason, we have to penetrate the global market,
Global mass.
especially the developed market.
Yeah.
Or the what we
US and Europe.
call as the regulatory market.
Right.
Yeah, so for that reason, we need to be clear, we have to have introduction of new technologies. We need to ensure that the quality of the products are right. And in that aspect, we need skilled men.
Killed man.
Because these are much complex.
Yeah, much
Of course, senior people, many of the places are actually trained abroad, they are coming back and doing it. But our bench level scientists and also the group leaders, managers, they also have to know this complexity of the biologics, otherwise how they decide when they are doing the experiments, is it correct or not?
Right, right.
And I think that is a challenge.
Right.
And my understanding from China and all these areas, I have noticed that there is a big difference in the number of skilled labors which are happening there. So that way I think ClinoSol is doing a good job. Look, it is not only preclinical, in clinical stage also, you understood, I told you how complex is
It is very complex, yeah.
So you need to have those people there. And another important thing, innovation.
Right.
Because any country, if they want to grow as a developed country, you need to have innovation in all areas.
Right.
You need to have innovation in pharma, you need to have innovation in IT, you need to have innovation in auto and all this, automobile and all these areas.
Right.
Why Europe and US are so far ahead?
Correct.
Because that's the hub of innovation.
Correct.
I see how China, Korea, Japan, Singapore are helping, government is helping, and how the academic and industry
Are collaborated. Right.
in that. Because see, end of the day, most of the initial discovery in those countries also happened in that country.
Right.
Then they transfer it to the industry,
Industries.
they take it there. In India, it is still a problem.
Yeah. there's a gap.
That has to be sorted out. Otherwise, innovation will be covered. And if you do not have innovative molecule, then you cannot reach that stage. But one thing I will tell you, a silver lining in this, is the area of cell therapy. In cell therapy, the first molecule which is approved, I think last month by CDSCO, Indian regulator, is from a company called ImmunoAct.
Okay.
It is in Mumbai just come.
Okay.
Now, if you look at its history, this actually started in IIT Mumbai.
Oh.
They discovered in IIT Mumbai.
Technology company.
Yeah. Then they worked with TIFR for the clinical trial. And when they found this, that this is what, and this is not a copy. This is an innovative product of
Innovative.
Yeah, in IIT Mumbai. Then the spin-off, what in abroad also they do.
Mm.
The cell therapy in abroad happened like that. That it was in UPenn, University of
Correct.
Pennsylvania.
Correct.
Then the spin it off, and then the
Correct.
I think Pfizer
Correct.
took it out.
Correct.
Okay. Now same thing happening here.
Huh.
So they spin off a company called ImmunoAct,
Yeah.
and that company then collaborating with the Lotus.
Yeah.
So Lotus, such
Yeah.
a big company,
Yeah.
they come into the field. They handhold them, and now they are working for developing this one for the larger one.
Larger one.
That means global one.
Right.
And you see how important this is for the global market also.
Global.
A cell therapy product in US and Europe
Hmm.
cost about 300 to 450,000 dollars
Mm.
for treatment.
Mm.
Whereas this one which is developed by ImmunoAct
Mm.
in India,
Mm.
it cost $50,000.
Yeah.
So you see how India is contributing
Right, right.
here, but this is not a copy.
Correct.
Like generally,
It's an innovative molecule.
This is an innovative molecule.
Right.
And there are also Immunil, which is a spin-off of Biocon,
Right.
and there are of course Stempeutics,
Right.
which is another company. But these are the who are working CAR-T,
Right.
Immunil and Immunac.
Right.
Whereas the one in Stempeutics who are working on stem cell,
Right.
they also have few products in the market.
Right, right.
So these are all innovative products. They are not copied.
Copy.
So
Yeah.
this is, as I said, silver lining.
The silver lining.
If really there are challenges.
Mhm.
You know, regulators, they have, I know, because I am part of those groups where we discuss these things, cell engineering one. There are regulators also took a lot of time to understand
It's
this.
the process itself. Right.
Because these are so new things. for
Yeah.
them.
Yeah.
Then there is a challenge of scale up.
Yeah.
See, cell therapy, you know, what happens is, you take the cells out of patient,
Right.
then outside the body you expand them,
Right.
pharmacologically treat them,
Right.
then do the whole analysis that they are good quality,
Right.
no contamination, nothing, put it back.
Right.
Okay? Now, there are two ways. One way is wherever you have patient, you do it there. That is called decentralized way of doing it.
Right. Decentralized.
Another is pull all
All
these
the
cells in one patient, one place. You can't pull patients. You have to take the cell and bring it.
Yeah. Yeah.
Okay? But that takes a lot of time,
Yeah.
a lot of money, and a lot of chance that when you bring this and all that, whether the cells will be okay or not,
No.
or the cells will get
Damage or
damaged.
something. Yeah.
But for that purpose, decentralization, you have to be very clear the quality you are achieving.
What?
If you achieve that, it is fine. Same is the clinical trial also.
Right.
Right now, only few patients you are taking in clinical trial.
Mm.
You need to have much more patients when you go in for the global
Larger population.
population. So
Right.
these are some challenges.
Some challenge.
And another challenge which I will say to talk, the last part of this talk is the raw materials.
Raw materials.
See, unfortunately, today in chemical medicine to some extent, we have raw materials in India.
Yeah.
Many of the raw
reactors and
materials.
everything.
But still, API is an issue.
Yeah.
And see, we saw during the COVID,
COVID.
and also this war,
Right.
that it put lot of pressure in the supply chain.
Right.
You know that, I mean, Serum Institute was not able to get the vials to
Vials.
put there. They have the vaccine.
Vaccines, but vials was missing.
What happened is vials come from abroad,
Yeah.
US or I think some European company.
Yeah.
Their government says first to give these vials to our manufacturers,
Yeah.
because we need the vaccine for
Right.
our population.
Right,
Okay?
right.
So you see how we get handicapped
Right,
in
right.
such a situation.
Right. Right.
I
Probably
think,
exists.
I think we need to have it, but I'm very happy. Just 2-3 months back, I had a meeting with DBT and also the Department of Pharmaceuticals, DOP. And they jointly now have focused this area. They understood that we need to really develop these raw materials.
Right.
They have put couple of schemes
Right.
where they are funding companies to start this kind of raw material production, both for chemical medicine as well as for biological medicine.
Right.
So I hope this will happen. Otherwise,
Right.
You never know where the globe is going. See,
Right.
one after another war is coming. And you see, these ships are not able to come through the sea.
Right.
Somebody is putting a missile and all that. So you never know
No.
what will happen at the end of the day.
Yeah.
So we must have our own production.
Production.
And
Raw materials.
without raw materials, you can't make anything.
Yes.
Whether it is a small molecule
Or a large molecule or whatever
so
it is.
this is something which is very important, and I am happy government has took note of it,
Right.
and they have started something. So, but one thing is there, as I told you that I think the Indian biopharma, both vaccines, I haven't talked so far anything on vaccine, maybe
Mhm.
another day I will talk about
Right,
that.
right.
And in biosimilar,
Right.
there is a big opportunity. They have to some extent proven them today to the global market that what is their potential. If they focus on the quality and have regular interaction with the regulators, I think we can take it like as we did small molecule, we can be a major player
For
outside.
the biosimilars and
And
biologics.
the reason I am very happy is government, including the Prime Minister himself, he realized the importance of this biologics or bioeconomy
Yeah.
overall. I mean, not only
Yeah.
for biosimilar or vaccine, but also in agriculture and that
Yeah,
there are a lot of
yeah,
things
yeah,
happening
yeah.
there. So he himself take a lot of interest and really figuring out that how exactly this can be
be
developed.
done.
He is exploring that how this can be funded.
Correct.
One challenge is today that still the funding is mostly from the government.
Government.
Some areas, as I said, Laura's and all that, the companies have stepped in.
Step
But venture capitalist funding is still,
little bit.
they are very open for IT and services. They will give and put money in it.
Yeah.
But in biopharma, they say, oh yeah, you know, it is very risky and all that. So there also, I think, no, but that's not in US.
Yeah.
They put a lot of money in
in-
US and Europe,
Yeah.
in the biopharma, biotech sector
Right.
and all that. So that also has to be.
Yeah.
Otherwise, to do a global reach, you need a lot of money.
Right.
DBT is not able to give you that.
Right, right.
You need this private venture capital.
Correct. So thank you, Ranjan sir. We had a lot of a nice discussion with starting with your career journey. To know about biologics and difference between biologics and new chemical entity, discovery of a biologics and what is biosimilars, what is interchangeable biosimilars and what is the other kind of biologic products or what are the new technologies and new innovations and what is the requirement for India to become a global market leader for all this. So we would like to thank you for giving so much of knowledge today.
Yeah, I think it is very important for the people who are aspiring to be in this field.
Yes.
Because see, chemical medicine to some extent now more or less filled up.
Filled up and saturated.
Yeah, these are the new areas
Yeah.
where people have to think about
about it.
So I hope I
Yeah.
could stimulate their thinking process
So I
with
think-
this information.
Yeah.
And I will be very glad. That's the reason actually
Yeah.
I have, because from USP I started.
Right.
I am consulting few biotech companies
Right.
who are starting this kind of activities
Yeah.
today,
Yeah.
and also teaching the students.
Students in
Who are putting this biopharma syllabus.
Right.
Because I think these two areas really they need guidance.
Right.
And that's what I put that, I will put my time on these, and if this helps to have few innovations
Right.
or more skilled manpower for the biopharma industry,
Right.
I will be very happy.
Right.
And I think all the things we discussed today,
Yeah.
so for the future generation,
Future generation and next
if they
generation.
feel excited on that,
Yeah.
and think this is an area they should come, then I think all our discussion is
That is very fruitful. I think we should have a couple of more discussion on this.
See, I
vaccine.
haven't gone some of the areas.
Yeah. So I think most interesting topic is vaccine. I think which our viewers will be very actively.
Oh, yeah.
Wanting
Because,
to listen to you.
even also today, biopharma, most of the ordinary people thinks is the vaccine. Because everybody taking it. And they knew so much rush there at the
Right,
initial.
right.
Now they are not taking that
Yeah,
much,
yeah.
which they should take.
Yeah, they should
Booster-
take. They should
booster
take.
dose.
Yeah.
But yeah, people know much of the vaccines, but not in the other areas.
Yeah. So thank you so much, sir, for spending today's time, and we'll meet for one more interactive session.
Sure. I'm glad I could do that. As I say,
Yeah.
that this is something which I have taken as a part of my post-retirement thing.
Right.
That I should encourage
the young generations to come into this.
and also see to it that they come here,
Yeah.
because otherwise we cannot really exploit this opportunity. What we have to do.
Right.
What you will do if you don't have skilled manpower?
Right. Perfect. Thank you so much for watching this, and we'll get back to you with one more video and one more information. Thank you so much for watching us.
Thank you.
ओके कुछ फोटोस తీసుకోండి सर कुछ तो बात करो सर वो पेपर रहने दीजिए सर कुछ तो बात करो सर you talk some natural things so that they take some photos for thumbnails hey what about తీసేనా
తీసేయండి
ఇప్పుడు చూడండి ఇప్పుడు ఓకేనా కొన్ని ఫోటోస్ తీసుకోండి natural గా सर कुछ तो बात करो सर you talk something आज दोपहर का खाना क्या बनेगा
No, I think it's
It's a very nice discussion, sir. आज कहाँ से पता मतलब हमने कुछ questions prepare नहीं किया. We will not discuss anything.
No, I think it will be good actually to have this kind of discussion, especially an organization like yours who are working with the students, as we said repeatedly, future generation.
Next
So
generation.
that is very important that we talk to them about these new areas and the new opportunities which brings them. And you know, many of the, see, as you told before this discussion, that one of the guys who used to work with us, he is now in GSK. So see, these are the areas. As of today, people in small molecule used to go and but now, even in bio area, if people have work experience in the Indian company, they have the opportunity to go abroad. Because now people know that. Like Serum Institute. You won't believe, from USP I have gone so many places across the globe. Everywhere they know Serum Institute. Because there's such a famous name in vaccine industry today. And many people from Russia, Thailand and other places, they ask me, can you please connect me to the Serum Institute? We want to collaborate and do something here. And they are doing it. And as far as I was possible for me to do that, I have done it. But so this is something is very important. And I am happy. The time we started in biologics, I told you, in Dr. Reddy, people used to laugh at us.
Basic money.
The people should, he said Dr. Reddy should put more on the API and or differentiated API, formulation, and all these. Because the drug discovery, NC research, cannot work with that biology. I said, why Dr. Reddy wasting money? From there today, it has come to a stage when the Prime Minister of the country is
talking
looking
about
this
biologics,
as something which can put India in the global
market.
And already we have achieved that with the vaccines.
Yes.
So that makes me very happy. That from those days, and also, you know, those days it was so difficult. That whenever you used to get some enzymes and all that for biology, people were only aware about the how to bring chemicals. So I used to tell them you have to bring it in ice. Then when it comes to my lab, I open the carton and see all ice melted. It is water where that enzyme was. And all the enzyme activity is gone. From there, today it has happened. Because now you have cold chain shipment where you have a monitor inside which gives you exact temperature at every hour. How it has been stored.
It's all technology, sir. It's all technology.
So all these changes have happened which has made us to work with biologics much easier in India.
Much easier. So one more update, done right? One more update, sir. We have that room, last room. We are putting that digital screen, so it will be so much big. So now when you come for next time, you will have a digital screen. You can put slides also. If you want to take live, you can take live on the slides, explaining the slides also.